to the next speaker, which will be Professor Willem Mulder from the Icahn School of Medicine at New York. And his talk will be about, again, monitoring and modulating innate immunity using nanomaterials.
Okay, let me see, is there a pointer? Is this thing, okay.
Yeah.
Okay, all right. Hey, so no, I understand. Okay, so in the first presentation, the focus was on adaptive immunity, on these T lymphocytes. This talk will be about innate immune cells and how innate immunity can be used as a target to modulate the immune system. So this story starts with the discovery of an innate immune memory, or a de facto innate immune memory, by Professor Mia Netea. We closely collaborate with Professor Netea. And so what he basically postulates is that cells, myeloid cells that are exposed to a microbe, they get epigenetically and metabolically rewired, and they are prone to have a more strongly response to secondary stimulus. And so this is through very different mechanisms, not through gene recombination, but it's through epigenetic modifications and metabolic rewiring. This is a de facto innate immune memory. And so this innate immune memory is regulated at the level of epigenetics, so these histone marks and cell metabolism, and that determines myeloid cell function. So these effects are durable, but they're not permanent. And so that's an important thing that has to be remembered. So a naive cell is exposed to a trigger, and we call it a training agent, that activates these cells. You get active gene expression, and then there's still persistence of histone marks, and upon re-stimulation of these cells after a resting period, the re-stimulation leads to a stronger inflammatory response. So this is at the level of individual cells. At the level of the immune system, this happens at the bone marrow and the bone marrow progenitor cells. And so there's some recent Cell papers that were published last January that kind of unraveled this whole mechanism. And so if this is a phenomenon that can be observed in vitro, so how does it work on the systems level? And that's because these changes also occur at the level of progenitor cells. And so the implication is that if you develop nanomaterials that accumulate in the bone marrow and associated with the progenitor cells, you can produce long-lasting therapeutic effects. So I'm not really going to talk about cancer, mostly not actually, but I think this is a very powerful paradigm. And it kind of represents two sides of the same immunological coin. So in transplantation, it's characterized by undesired immunity, and that results in T-cell activation and rejection of an organ, so that requires immunosuppression. In cancer, kind of the opposite holds true. So there's tolerance by the immune system towards a tumor. And so if we look at the interplay between, let's say, antigen-presenting cells, the myeloid cells, innate immune cells, and T cells, and you see that antigen presentation is required, as was shown in the first presentation, co-stimulation, so there's also these checkpoint proteins, and soluble cytokines. And these three signals cause the activation of T cells. So we don't want to block any of this, and we don't want to focus on the T cell. We want to be able to regulate this at the level of myeloid cells, so at the level of innate immune cells. And therefore, we focused on this concept of trained immunity, because that's a de facto innate immune memory of these myeloid cells. And so if you regulate this trained immunity in these cells, we should be able to regulate the immune system. For that, we've developed nanotechnology. My lab has been working for more than 10 years on endogenous materials, lipoproteins. And so lipoproteins transport fat throughout our body. A very famous one, or well-known one, is high-density lipoproteins, also known as good cholesterol. and it has inherent features that it contains these phospholipids and other lipids, and apolipoprotein that interacts with ABC1 scavenger receptors and other immune cell receptors, but these are receptors that are expressed on innate immune cells. So then we extract high-density lipoprotein from human plasma. We can also extract components or create components through recombinant DNA technology. But this, in essence, is what we do. I mean, we skip this step. So we collect human plasma that we get from the blood bank. Then we isolate the high-density lipoprotein fraction. And so we do through extensive centrifugation steps. And then we basically can disassemble high-density lipoprotein and reassemble it with a payload. And it can be a therapeutic payload. We can put in polymers. And that allows us, using microfluidics technology, to create these high-density, we call them HDL nanobiologic libraries. And so these are not real high-density lipoprotein particles because we've been playing with them too much, but we call them nanobiologics because they're based on high-density lipoprotein. And so we can make them really small and discodal. So you see here, they're about 10 nanometers. We can grow them in size by the incorporation of triglycerides. And they're about 30 nanometers in diameter, all the way up to 400 nanometers by the inclusion of polymers. And by means of imaging and flow cytometry, we can screen these libraries for specificity for immune cells. And so this is the first time I present these data. This is a paper that we got very recently accepted in Immunity. And so we apply this paradigm in the context of transplantation. One of the regulators of trained immunity is the mTOR pathway. So what we've done here is we created a nanobiologic that contains an mTOR inhibitor in its core. So these things are about 20 nanometers in diameter. And in vitro, we see that these things very effectively inhibit trained immunity. So in red, it's basically naive cells that are exposed to LPS, which is the secondary stimulus. These are cells that are trained with beta-glucan, so that's a trainer, or that's basically stimulant of trained immunity. And when we co-incubate with our nanobiologic, then we basically see what you see here in black. So we prevent this trained immunity phenotype on human monocytes in vitro. Then we've done some work and found that these trained immunity mechanism also seems to occur, at least the trained immunity-like and associated processes in transplantation. And so then we designed a study where we first evaluate this nanobiologic containing the mTOR inhibitor that we label with either radioisotopes or with a fluorophore. And then we look at biodistribution and immune cell specificity. But what's very important to note here is that there's a lot of uptake in the bone marrow here. And that's where our target is. That's where trained immunity on the systems level occurs. And then through flow sorting, we also looked at the myeloid cells in the bone marrow. You see the lymphocytes don't take up our material. It's purely the myeloid cells and the progenitors of a whole bunch of different myeloid cells. Now, the transplantation model is a model where we have a C57 black 6 mouse, that's really a black mouse, that receives a heart from a BALB-C, it's a white mouse, and it goes into the abdomen of the mouse, so it receives a second heart, which is connected to the abdominal aorta. It's not functional, but it's beating. And this model typically rejects at day 8. And it's a very reproducible model. And you feel that through palpation. Basically, the heart stops beating. The animal doesn't die because it's not functional, but it stops beating. Then in the field, it's considered that if you take this all the way to 100 days, and so apply therapy, that's considered tolerance because then the hearts shrink and they basically merge with surrounding tissue. So what we've done here is a very mild regimen where we infused at day 0, at day 2, and at day 5. So before the heart would normally reject, we inject these infusion of we inject the nanobiologic, and that results in a rebalancing of the immune system. So we see that the ratio of these Ly6E high, which are the inflammatory monocytes, to low monocytes, changes dramatically. And then when we isolate through flow sorting monocytes or myeloid cells from the allograft, we see that the mTOR pathway is downregulated. And the mTOR pathway is metabolic pathway that's highly relevant in trained immunity. So how does this influence T cell function? And so we reduce basically the co-stimulation and the cytokine production of these myeloid cells. And so now we want to see is like how that influences T-cell immunity and T-cell activation. So we've done an experiment where we first see that the Ly6C low population goes up, and so these are the less inflammatory monocytes in the allograft. We flow sort these cells, and then we co-incubate these cells with T cells, and what you see is that the proliferative capacity of these T cells goes down. So they become less active because they're in the presence of treated myeloid cells that are in vivo treated. And then what we also see, there's higher FOXP3, so there's more regulatory T cells. And that's what you want in the context of transplantation. So then we had to prove that it's really myeloid cell specific. So we use a model where we can deplete the Ly6C low population. And then this is our survival curves, allograft survival curves, where you see that the therapy doesn't work. And we can recover the therapeutic efficacy by the infusion of monocytes. And so it is really monocyte or myeloid cell dependent. So that's what we've shown with this specific experiment. But then what it's all about, so how well does this work? I told you 100 days is considered tolerance. And so these are control groups, so we have placebo, and we have a vehicle nanobiologic, and you can nicely see that you have dramatic prolongation of allograft survival. And remember again, it's just an infusion at day 0, 2, and 5, no subsequent immunosuppression. So we thought this is pretty spectacular, but we would like to get them all to 100. This is a paper that we published earlier this year, in which we target TREF6 with also a nanobiologic. I won't go into the details, but this one also targets myeloid cells, but then prevents CD40 ligand signaling, which is the co-stimulation. So we thought, like, well, let's see how well that works in transplantation. That's what you see here. And so it doesn't work as well as the trained immunity mTOR specific nanobiologic, but it also has prolongation. But then when you combine both of them, you get almost exclusive tolerance. And so you see that most of the animals, they have hearts that survive up to 100 days. So our nanobiologic therapy specific to mTOR, we've also applied in the context of cardiovascular disease. I won't go over all the details, but it also works in atherosclerosis, where you also want to basically dampen inflammation, and we see a nice reduction. And this is 1 week of treatment. So these are animals that have advanced atherosclerosis. We treat them for 1 week. They get 4 infusions and see a really dramatic reduction in inflammation. So this is transplantation. In cancer, you want to do the exact opposite. You don't want to dampen trained immunity. You actually want to induce trained immunity. This is an ongoing study that we're about to collect. We've collected most of the data. Looks very spectacular, but for reasons of IP and other issues, I can't disclose too much and not too many details. I just want to show you how well this works. So this is B16F10 melanoma model, which is actually notoriously hard to treat with immunotherapy. But we see that we can almost completely halt tumor growth in this model by the induction of trained immunity. So in summary, we like to use nanomaterials to regulate the immune response at the level of innate immune cells. Imaging is always an integral part of what we're doing because we really want to understand where these materials are going. We need to be able to screen, and we need to be able to understand how they behave in vivo. And we've also translated these technologies to larger animal models, to rabbits, to pigs, and even to non-human primates. And so then imaging becomes very, very important because we can't process 300 monkeys. But with imaging, you can reduce the number of animals. And then we have a lot of strong focus on cardiovascular disease, this transplantation work I've just shown, and malignancies. And then I'd like to end this talk with thanking you for your attention and acknowledging my collaborators and funding. Thank you.
Thank you very much, Willem. Great talk. Questions?
Nice presentation. Is there anything unusual about the metabolism of the APOA1 and these HDL particles in the muscles or in the liver? Any like safety studies you've done with these animals?
Yeah, so this actually was this paper, if you'd like to see it. But so we've done very extensive tox work in this paper in mice and non-human primates, and we basically see it's safe. We don't see any differences in, they say, liver markers. We don't see a huge impact on the immune system and white blood cell count and all these things. So seems to be pretty safe. But this is one formulation, and we work with other formulations for which we've not done extensive talks.
Very interesting talk. How do you explain that only 3 infusion causes this immune tolerance? What do you think?
Yes, so the paper will be out soon, and I'm happy to send it to you. But the problem starts at the onset, so at the transplantation itself. It basically induces an inflammatory response that activates the immune system, and that causes downstream problems. And the current treatment paradigm is that then you treat these symptoms, which is, you try to deactivate T cells. So what we do is we prevent these initial detrimental inflammatory effects, that, keep them down, which then doesn't result in T cell activation and T cell immunity.
So, I mean, the big advantage of transplantation is that you can pre-treat the recipient. And so did you actually also test that you would already give 1 week before transplanting the recipient your infusion, because you may even get better than where you are now?
Yeah, so that's part of our ongoing program, because that's the most obvious thing to do, that you do before. So we do it at the time of transplantation. When we put the animal down, it receives simultaneously. So this is more because of logistical reasons, and obviously when you start something, you don't know it's going to work that well, and then you have to stick to that regimen. But it makes more sense to do it a couple of hours prior or even a day before. Yeah.
More questions? Yes, Dan?
Thanks. What about the loading efficiency of the particles?
Yeah, so we have very nice tricks to get that very efficient. And I'm happy to discuss that with you outside the context of this thing, because there's also, but we have basically developed a way to, in a modular fashion, incorporate small molecule drugs, and then with efficiency of 80 to 90%. And it's across the board. It doesn't matter what the drug is. I mean, it matters to some extent, but if you just focus on small molecules, so you think about glycolysis inhibitors, in this case we use rapamycin, which is not typically a small molecule, it's actually a pretty large molecule, and so we can get incorporation efficiencies of about, well, maybe incorporation efficiency is the wrong word, but we can get drug recovery of about 80 to 90%.
So you have efficiently tolerized your system in the allo heart transplantation system. Can you re-induce immunity in this system?
Yeah, that's a very good point, and that's also what we've done as mechanistic studies. So we also try to see if then we can induce rejection again, and that happens.
So that's the answer why priming does work, and vaccines may work, because you can also re-immunize and tolerize.
Yeah, and I want to emphasize again, so this trained immunity is based on epigenetic and metabolic rewiring, which is long-lasting but not permanent. And so in cancers, you think you can induce trained immunity, and then when the cancer is resolved, the immune system goes back to a normal immune system, so you're not as susceptible to all kinds of infections and all kinds of other problems.
Good. Last chance for a last question. If not, let's thank Willem again.